missing. Does anyone need this to get signed in for the rest of the semester, so you don't have to fool with it next week when you're testing? And again, get your notes open to this, exactly where we left. And I said we'd be starting with, how does that sodium get in there? So you have the pictures in front of you. There's no reason for me to redraw the whole thing. I'm just going to put a little bit of it up here. And we call this depolarization or upswing. The influx of sodium. And so how's the sodium getting in there? So I need you to picture the cell membrane as you studied it in biology. Bilayer phospholipids with proteins dispersed throughout the phospholipid bilayer. These proteins were of many types. Some created little hydrophilic channels. Others were active transport systems like the sodium-potassium pump. Some of the proteins were on the inner surface of the membrane and worked as enzymes. Others were on the outer surface of the membrane and worked as receptors. These could be receptors for hormones or receptors for your immune system to know you versus not you. Lots and lots and lots of examples of those membrane-bound proteins. Well, now you're picturing some membrane-bound proteins that work as gates, just like the gate in a fence. The gate can open, the gate can close. So this influx of sodium is through sodium gates. There are two types of sodium gates. Again, these are membrane-bound proteins that are like the gates in a fence. They can open or they can close. So this influx of sodium that was responsible for taking us from -70 to +40 is through gates. First, chemical gates. I'm using the word chemical very loosely. It does not have to be a solution in a bottle. You've also got the graph in front of you from Monday. The chemical gate is a gate that opens in response to an environmental stimulus. And I'll give you some examples now, but it's something that will be self-clarifying as we proceed, not just today, but after the holiday season. So for example, when I touch this, and you know, we've done this a million times now, but when I touch this and I perceive this as rough or smooth or hot or cold, the receptors in my fingertips are being stimulated by this, and those receptors are actually chemical gates. And sodium moves in through those chemical gates. If you stick out your tongue and I drop sugar water on it, or vinegar on it, or salt water on it, that liquid material will open a chemical gate, and sodium will begin moving in through that chemical gate. When I look and I see you, light is shining into my eye, interacting with a structure you've all heard of. You don't have to know anything about it, but the retina. The light stimulates the retina and opens chemical gates, and sodium moves in through those chemical gates. When you speak and I hear you, the sound, which is just moving air, by the way. So when you speak, you make the air move. The moving air makes my eardrum move, which makes hair cells move, which opens chemical gates. The opening of the chemical gates either does or doesn't take us to threshold. And that's why on Monday I used a dash line. You know, I've got negative 10, 20, 30, etc. down to negative 70. Why did I throw in negative 55? It is the threshold. So the sodium moves in initially through chemical gates. If threshold is reached, which is the negative 55 millivolts, the second type of gate opens. The voltage regulated gates. So when this environmental stimulus or molecular stimulus in the body, whatever it is, when it occurs, all it has to do is take me to negative 55. If I reach negative 55, then the voltage regulated gates open and an action potential occurs. If this does not take me to negative 55, I return to rest. It is a threshold. It is a phenomena known as all or none. There is no such thing as a strong action potential, or a weak action potential, or a kind of sort of action potential. You reach threshold or you don't. It is like earning an A. 899 points is not an A. 900 points is an A. The line has to be drawn. There is a line between the state of Colorado and the state of Kansas or New Mexico. No, you can't have an extra millimeter. This is the line. It is absolute. It is all or none. It is like these lights. They are on or off. There is not a dimmer switch here. Now at home, in some of the rooms, wherever you live, you probably have dimmer switches. There's not a dimmer switch here. These lights are on or off. Yes or no. Binary, 1 or 0. Have I made my point?
Yes.
I'd like to continue. So, even if you take this into the laboratory and you stimulate it to negative 40, it doesn't change what happens. You reached threshold. If you stimulate it to negative 56, do you get an action potential?
Yes.
No, because that's not negative 55. Get your numbers in order. Negative 56 is below negative 55, so you don't get an action potential. If you stimulate it in the lab with an electrical current all the way to 0, you just get an action potential. You don't get a better one or a stronger one. It happens or it doesn't. Yes or no, all or none. So this influx of sodium from Monday is via sodium gates, proteins in the membrane that open and close. The first type of gate opens in response to some chemical stimulus, using that word very, very loosely. And if we reach threshold, then the voltage regulated gates open. Now, you've still got your picture in front of you. So after this picture, your next picture showed the depolarized cell, and it is sitting there at positive 40 millivolts, and it has high potassium, high sodium, high anions. Extracellularly, the potassium is low, the sodium is low, the anions are low. And what did we call this next part of the action potential?
Polarization.
Repolarization, exactly. Or downswing. And this was potassium moving out. Well, this side is simpler. It's still gates. These are voltage regulated potassium gates. So it's simpler because there's just one type of gate here. We've already reached threshold. Okay, there is no threshold on the downswing side. So if depolarization occurs, repolarization occurs. So the all or none doesn't pertain to just depolarization, it pertains to the action potential. All right, looking at Roman numeral 4 on page 36, we've got 6 things there to discuss in the context of action potentials. Looking at your pictures from Monday, what had to happen after repolarization before the cell could undergo another action potential? Had to recover, because our sodium and potassium were in the wrong places. So what I want you to think about is, does the cell have to recover? Might there be exceptions to that? And the answer is, well, it depends. The inability to undergo another action potential is known as refractory. So the refractory period would be when the event can't occur. And it doesn't have to be action potentials. Refractory period will come up next semester in the context of the reproductive system. So as you see on your notes, you have 2 types of refractory. Absolute refractory, it is absolute. It's impossible to begin another action potential. The sodium gates are already open. The cell is depolarizing. This one is so simple, it's stupid. But it's science and everything has to be defined. This is the same as me asking you, so I can open the door, I can close the door. If I am opening the door, as I am now, is it possible for me to begin opening the door? Dumb, isn't it? I'm already opening it. How can I start opening it? That's what absolute refractory is. If we're depolarizing, can I start depolarizing again? Stupid. But it's science. Everything has to be defined. The other type of refractory, relative refractory. The cell has completed depolarizing. And has started repolarizing. So that is, the sodium gates have closed and the potassium gates have opened. So in this case, the question is, if I finish depolarizing and I've started repolarizing, I wonder if I could skip recovery and begin depolarizing again. In other words, could I do this? So I didn't finish repolarizing. Or could I do this? I almost finished repolarizing. Or could I come all the way to the bottom but skip recovery and depolarize again? Or could I stay up there for a little while? Maybe. Maybe. There are two requirements for this. A stronger than normal stimulus and secondly, I've got to have some extracellular sodium. That's what depolarization requires, is the influx of sodium. If there's zero extracellular sodium, it ain't going to happen. So absolute refractory, we are somewhere on the depolarizing side of the curve. It is impossible to begin depolarizing if you are currently depolarizing. But in relative refractory, you have finished depolarizing, and the question is, maybe I don't have to go all the way to the bottom before I go back up. Maybe I'd like to go to the bottom and get the food out of my car and eat it before I go back up, but maybe I don't have to. Maybe I could just go halfway down and turn around and go back up and burn some fat. Now the stronger than normal stimulus, I don't know, let your imagination run. Somebody's chasing you, and if they catch you, they're going to do things so horrible that you're going to beg them to kill you instead. You're going to run some sub-3-minute miles. Another stronger than normal stimulus. This is a lip. You can see my fingerprints here from the years. That tells you how often they clean the room. But this is a lip, and I'm hanging over the edge of the Grand Canyon. How long can I hang there? Not very long. Or maybe until you go get a rope to save me. Because if I slip and fall, I'm going to plummet 4,000 to 5,000 feet to the river. Stronger than normal stimulus. Let your imagination run with that one. All right, our next couple of terms, concepts, ideas, letters C and D, cable properties and saltatory conduction. So to talk about that, we need to go back to a picture. Either one of these will do, but I guess the one on the left is a little better. So remember that the nerve cell process is myelinated. Is this PNS or CNS? How do you know it's P? Swan cell. If it was CNS, the cells would not be swan, they would be oligodendrocytes, yeah. So the nerve cell process is myelinated via the swan cells. I suggest that I have enough trouble convincing you that all this action potential stuff even happens. Maybe I just made it up. No, of course not. But it's kind of bizarre, right? And the last thing we did on Monday was see how, while it took 10, 15, 30 minutes, I don't know to explain it, how many times does it happen between the time I touch this and the time I feel it? Anybody remember? 914 times? What took me half an hour to explain just happened 914 times? Wow. So it's hard enough to convince you that any of this is even real, but there's no way in the world I could convince you that the sodium and potassium move through the swan cell. So where along the nerve cell process do you suppose the action potentials occur?
Nodes.
At the nodes. At the nodes, and only at the nodes. So the action potentials jump from node to node. Translation, saltatory conduction. The action potentials jump from node to node. So somebody says, what's saltatory conduction mean? And you say, that means the action potentials jump from node to node. So I'm going to wrap this axon with swan cells, which of course are perpendicular to the plane of the board. There's a swan cell, another swan cell, another swan cell, and so on. Saltatory conduction, the action potentials jump. Not literally jump, but appear to jump from node to node. If an action potential is occurring here, meaning sodium is moving in right there, and you apply what you learned about diffusion, high concentration, low concentration, does that sodium just sit there? It'll spread out. It'll spread out. Now, before any of that sodium moves, what is the voltage differential right here? And as sodium begins to diffuse toward that node, what does that do to that voltage differential? Raises the voltage. And what happens when enough sodium diffuses to that node to change the voltage differential there from negative 70 to negative 55? It goes bang! Not really, but you get the point. What happens here when we reach negative 55? The voltage regulated gates open. So when the sodium, enough of it diffuses to the next node, and the next node reaches threshold, the voltage regulated gates open there, and sodium begins to diffuse into that node. It doesn't just sit there, it diffuses toward the next node. And when enough sodium diffuses toward that node to depolarize it to threshold, the voltage regulated gates open there. So the action potentials occur only at the nodes because there's no way that ions can move through the myelinated regions. And each action potential stimulates the next one. Why does an action potential occur here? Because the preceding action potential occurred. So they jump from node to node. That's called saltatory conduction. Now, how come they jump from node to node? Because the sodium, what's the word? Diffuses. That's known as the cable properties of sodium. Cable properties define. Sodium diffuses from node to node. Cable properties define. Sodium diffuses from node to node. This results in saltatory conduction. The action potentials occur at the nodes. Each action potential stimulates the next one. So the all or none law now all or none does not pertain just to whether or not we reach threshold and depolarization occurs. If we reach threshold, yes, depolarization occurs. But if depolarization occurs, then repolarization occurs. But beyond that, all or none pertains to this entire axon. If we get this action potential, we will get this one. And if we get this one, we will get this one. Why? Because the sodium diffuses from node to node. And that's called cable properties. And their jumping from node to node is called saltatory conduction. Now I wonder what determines whether or not we get the first action potential. Because if we don't get this one, we won't get that one. So there are some other words right there on your notes. Letters E and F, I suppose, but I'm guessing. Summation, spatial and temporal summation. So we have an abbreviated version of a neuron. Looks like this. And when we talked about neurons, we said that dendrites, which are the dendrites? These little short ones or the long ones? Dendrites conduct signals toward the cell body, and axons conduct signals away from the cell bodies. Okay, so the question is, does that axon fire? It's resting at -70. What do we have to get this axon to for it to fire? -55. Okay? To make the math mathless, we will use units of 5. So let's suppose, well, let's get our drawing complete. So I have several upstream neurons connected to the downstream neuron. All right, let's imagine that this upstream neuron fires 3 times rapidly. Bang, bang, bang. And each time it carries a value of 5. So it fires value of 5. Before it returns to rest, it fires again a value of 5. Before it returns to rest, it fires again a value of 5. What happens here? I've got positive 5 times 3, so what do I have here? Negative 55. So, action potential occurred. That is summation over time. This neuron fired, whichever one I said, 3 times. Bang, bang, bang. Summation over time. You got 2 words there. Which one means time?
Temporal.
There you go. So that is temporal summation. Now I want you to imagine another scenario. We're resting at negative 70. And simultaneously, this neuron fires, and this one fires, and this one fires. Zap! All 3 of them fire instantaneously. At the same time, so there's no summation over time, but these are in different places. We got 2 words there to pick from. What type of summation is this? Spatial summation. Spatial summation. Another scenario. This one fires, and simultaneously that one fires, and then this one fires again. What type of summation is that? Yeah, exactly, both. These 2 fired simultaneously. They're in different places, so that's spatial, but then this one fired a second time. So that the temporal is in there too. So we don't have to have just spatial or just temporal. We can have the 2 together. Oh, let's do something crazy here. What if this one fires? Well, let's say these 3 fire simultaneously, a value of 5. And at the exact same time, that one fires but carries a value of negative 5. What's that do to my minus 70? Makes my minus 70 minus 60. So I didn't reach threshold. So does this cell fire? No. Now I want you to imagine trillions of these in your brain. And this is what is happening when you move, or when you make decisions, when you feel love versus hate. Everything comes down to this. And this is what we try to replicate right here. And we use transistors. Some of you know a lot more about what I'm talking about than I do. But I remember when my father showed me a transistor and I was a little boy, back in the 40s. And it was as big as a granddaddy long-legged spider. And he said, son, one day we'll be able to put 10,000 of these on the head of a pin. Whatever, Dad, I'm hungry. Could we just go to Mr. Steak and eat? And we did. How many millions of transistors can we put on the head of a pin today? A lot, yeah, a lot. And that's what makes these things work. And as cool and as fast as these things have become, the smartest, fastest computer in the world is stupid compared to a fruit fly. That's how fast and intelligent this stuff is. Trillions and trillions and trillions of these in our brains. And when you take drugs that make you feel weird, whether it's good or bad or whatever, you're affecting what happens at these synapses. Because we have, as you can see here, excitatory signals and inhibitory signals. Let's look at one more scenario because there's a word I need to define. So we get, I don't care what numbers we use, we'll stick with values of 5 to make the math mathless, but I want this number to get smaller. So let's say we go +5, +5, -5, -5, -5, all simultaneously. So what's that do to this number? -75. So we're resting at -70, and we go down. When we were at -70, how far away from threshold were we? 15. Now how far away are we from threshold? 20. What's the word that means there's a voltage differential that starts with a P?
Potential? No.
Polarity? So are we polar here at -70? The only place we're not polar is at 0, right? So we're polar at -70. Well, now at -75, are we more polar? So what happened here is called hyperpolarization. Hyperpolarization. Translation, we're further from threshold. Have you ever been trying to make a decision and you go back and forth? This is what's happening. Have you ever been brushing your teeth and you almost gag? Don't lie, everybody's done that. But you don't. But you almost, but you don't. But you, this is what's happened. This is exactly what's happened. Now once again, I've said this several times, but it makes the point of just how bizarre life is. You pick your sport, figure skater, gymnast, diver. Would you imagine that person coming off the board, the beam, throwing themselves into the air with their figure skates, and how hundreds and hundreds and hundreds of muscles have to contract. Every muscle has to contract in the proper order, and each muscle with the exact amount of strength. Otherwise, they land on their... And we felt computers were cool. All right, our next topic . What do we call this space?
Synapse.
This is Roman numeral 5.
Do we need a fresh page?
Oh, yeah.
Okay.
3 duct taped together.
Okay.
I have a little preliminary information before you need the fresh page. I'll caution you. So, and as you know from a week or 2 or 3 ago, special chemicals called neurotransmitters move that signal. You know that if this second cell, instead of being a second neuron, the second cell is a muscle, this is called a myoneural junction. In other words, molecular events of muscle contraction, action potential in a motor neuron, release of what at the myoneural junction? Acetylcholine. You know that acetylcholine is the most common neurotransmitter in the body. Abbreviated ACH. There are other neurotransmitters in the body. Dopamine, norepinephrine, serotonin. Those are the four we'll talk about. We won't necessarily talk, well, we definitely won't talk about all of them today. If we don't get to serotonin today, I'll remember next semester that I didn't cover it before the break, and I need to cover it after the break. I definitely will not cover these today, other than to say collectively, these are the big three in terms of why does methamphetamine, why does heroin, why does pot, why does ecstasy, why does alcohol make us feel the way they make us feel? Those are the big three when it comes to mood altering. We will, and I don't know if it'll be today or not, but this one's fascinating to talk about, and I certainly don't want a show of hands, but some of you know someone, or perhaps yourself, who takes a medication for depression. Drugs like Paxil, Zoloft, Wellbutrin, Zyban, Effexor, and it's those drugs act on serotonin. If your serotonin level is low, you feel like crap. And the reason you feel better in the summer than in the winter is because your serotonin level is higher in the summer. In fact, right now you feel a little crappier every day, because every day is a little shorter. And so you feel a little crappier every day. And if you didn't, now you will, because I've suggested it. And this will continue until December 21st, the shortest day of the year. Starting December 22nd, you'll start feeling a little better every day. And you'll max out on June 21st, the longest day of the year. And we know this is true because we plot things like suicide rates, admissions into drug and alcohol rehabilitation facilities, weddings, prescriptions written for selective serotonin reuptake inhibitors, drugs like Zyban, Wellbutrin, Zoloft, Paxil, Effexor, blah, blah, blah. Anyway, so it's cool stuff. And the most powerful selective serotonin reuptake inhibitor in the world is MDMA, which is what the young people in the room may know as ecstasy. The thing is, ecstasy is a bunch of crap mixed with it. You might be interested to know that up until 1984, ecstasy was legal. Just went in a bar and bought it. Nobody thought anything about it. It was fine. Nobody cared. And then the military realized it made people feel good. They said, we can't have that. So the government outlawed it. True story, 1984. We played checkers that day, I remember. Remember the bar where we bought it. We didn't do it, we just wanted to see that it was possible. Clean sheet of paper turned sideways. And it was not ecstasy that people were buying back in the 70s and 80s. It was pure MDMA. One more time, and don't draw this because you'll just have to erase it. So this is what we're looking at. So we're going to have a huge picture of just this, okay? Doesn't matter whether it's nerve, muscle, nerve, gland. The most common neurotransmitter is acetylcholine. This is called axon terminus. These are synaptic vesicles. Filled with neurotransmitter.
Bless you.
And in this case, the particular neurotransmitter is acetylcholine. This is the downstream neuron, or the muscle, or the gland. Does not matter. Once again, acetylcholine is the most common neurotransmitter in the body. So we could be stimulating a muscle, as in molecular events of contraction. We could be stimulating the parotid gland that you're familiar with in the cadaver. You know it makes salivary amylase. We could be stimulating a second neuron. On this downstream cell, we have receptors . These are receptors for acetylcholine. These receptors contain chemical gates. Whole gate thing again. Influx of sodium is through gates, two types of gates. First type of gates are chemical gates. They don't let a lot of sodium in. They do or do not let enough in to depolarize the cell to threshold. If the cell reaches threshold, then the voltage regulated gates open. These receptors contain chemical gates. The green thing is an enzyme called acetylcholine esterase. Digest that word, acetylcholine esterase. ACE tells you the word is an enzyme. What is this enzyme going to break down? Acetylcholine. It breaks down acetylcholine almost as fast as it binds. Why would it do that? To prevent continuous stimulation. To prevent continuous stimulation. All right, now we have our picture. We can talk about it. Try to give it steps, like we did with the molecular events of contraction. So the action potential reaches the axon terminus. So, we get the influx of calcium. So out here, calcium is at a high concentration and it moves into the axon terminus. This causes the synaptic vesicles fuse with the presynaptic membrane. So all of these synaptic vesicles fuse with the synaptic membrane. You draw from Biology 111. What's going to happen next? What goes on between plasma membranes and vesicles? Two E words. Endo and exocytosis are the two E words. If that vesicle just moved over and fused with that membrane, what's going to happen next? Hope you didn't write with a pen. Oh crap, wrong color. So what happens next is the exocytotic release of acetylcholine. Now remember, all of these vesicles have done that. I'm just drawing sort of in time lapse. You can, if you're interested, Google this stuff and find really cool little animations, YouTube or whatever. A lot of people enjoy making animations like this. Well, the acetylcholine binds the receptors on the postsynaptic membrane. This binding opens chemical gates for sodium. Well, if we've opened chemical gates for sodium, we've got sodium moving in to that cell. Sodium begins to move into the downstream cell. What's the voltage differential in this downstream cell before any sodium moves into it? Negative 70. What's happening here as sodium moves in? Negative 69, negative 68, negative 67, negative. Enough sodium does or does not enter the secondary cell to depolarize it to threshold. So if you're wondering, well, why might it not? Well, what if we didn't release enough acetylcholine? What if by not releasing enough acetylcholine, we didn't activate enough receptors? If we didn't activate enough receptors, maybe we didn't open enough chemical gates. Okay. So maybe it happens, maybe it doesn't. Enough sodium does it enter secondary cell to depolarize the threshold. If the secondary cell reaches threshold, read my own writing sometimes. Cry when I think about you poor people having to try to read it. If the secondary cell reaches threshold, the voltage regulated gates open and the downstream cell is stimulated. That is, an action potential occurs. Again, I want you to think about this because it's something we'll build on. It probably won't be until after the break. But, you know, what could be going on if we don't get this cell to threshold? All this stuff happened, but this cell reaches negative 56 or negative 60 only. Maybe we didn't release enough neurotransmitter. If we didn't release enough neurotransmitter, we didn't bind enough receptors. We didn't open enough chemical gates. Maybe we released enough neurotransmitter, but maybe this cell doesn't have enough receptors. Or maybe we released enough neurotransmitter, but the neurotransmitter was broken down before it could bind the receptors. Okay? So all physiology and all pathophysiology and all drug action has a molecular basis. And that's why we go into all of this detail, so we can truly understand things. I remind you that the Egyptians got it wrong, with no concept of any of this. They cut people open and said, well, I bet this does that, and I bet that does something else. And they thought the brain was garbage and ripped it out through the nose. They thought it was all about the heart. Well, that was kind of silly when we look back on it. And then for decades, for centuries, we thought that the heart pumped air and liquid. I don't remember which we thought was which, but we thought, so I'll just make it up, we thought the arteries carried liquid and the veins carried gas. And that could be backwards. Like I say, I don't remember. That was just too much to keep up with. But we were crazy. That's stupid. We know now the blood vessels all carry liquid. It's called blood. Some of the blood is oxygenated, some is deoxygenated, but none of the blood vessels carry air. The lungs are involved in that. But see, without microscopes, without imaging, without an understanding of the periodic table, how could we ever figure any of this stuff out? All right, bringing this to a close, and it's basically what I said somewhere right there, to prevent continuous stimulation, acetylcholine esterase hydrolyzes acetylcholine as fast as it is released. Now, I could probably do it, but I'm not going to try to do it in 9 minutes. So as far as covering serotonin, that'll be where we begin after the break. But I've got to say, as I've said before, guys, when we come back from the holiday break, it's got to be like we've been on a 5-minute coffee break. Okay? And I don't know if you're in the room, but I know at least one of you is taking micro and something else in the spring and taking 202 in the summer. The story's the same. When we come back in the summer, it's got to be like we've been on a 5-minute coffee break. So we'll pick up with serotonin. But I want to talk about it a little bit, even though I'm not going to test you on it. And any questions that are on the exam will just be thrown out, because I typically do talk about serotonin today. But I've given you various reasons for this. You know, enough sodium does or does not enter the secondary cell to depolarize it to threshold. Well, let's imagine that the neurotransmitter here is serotonin. And guess what is downstream, or guess what is at the distalmost end of this pathway if we're talking about serotonin? Pleasure centers. And it's not that we're trying to stimulate our pleasure centers so we feel all good. It's that if we don't stimulate our pleasure centers, we feel bad. I mean, people suffer depression for a number of reasons. Pull into your garage and run over the cat. You're going to be sad, but you're going to get over it. I mean, both my parents are dead. I was very sad. Holidays are always difficult. But life goes on. You know, I'm a happy person. I don't dwell on it. I'm not depressed anymore. I was. But life goes on. And you've all experienced traumatic events. And life goes on. But sometimes people feel bad. Sometimes people have the blues. And they don't get over it. And it could have started with a horrible event like the running over the cat or the loss of a parent. But they don't get over it. And sometimes we don't know why it starts. The person is seeing their doctor just for something routine. Maybe they have medication they take for their cholesterol or their blood pressure, or maybe it's a pelvic exam, you know, and the doctor's just making up, well, how have you been? You know, now that you mentioned it, I've kind of felt like crap. Really? Anything going on? No, the job is great. The children are doing great in school. Well, what? So you may not even know what causes the depression. So what's happening, though, is these pleasure centers aren't getting stimulated. And so we think that sometimes it has to do with back at these synapses, but instead of acetylcholine, the neurotransmitter is What are we talking? Serotonin. Serotonin. So some of these reasons that I've already given for why might the secondary cell not reach threshold. Maybe the person doesn't make enough serotonin. Maybe they make it, but they don't release it. Maybe they make it and they release it, but there aren't enough receptors here. Maybe their body's attacking the receptors. Maybe they make it and they release it, but they enzymatically break it down before it has a chance to bind. We're not going to know which it is, but the drug that you try is a drug designed to keep the serotonin in the synapse longer. If you keep the serotonin in the synapse longer, you increase the chance that binding occurs and downstream stimulation occurs. And there's so many of them out there. I've rattled off a bunch of names. I won't do it again, but it's just almost a crapshoot as to which one your doctor tries first. He or she will probably try the one that he or she has had the best results with. If he or she doesn't know, then it's just like talking about anti-inflammatories. Let's see, which one lands closer to you? That's the one you'll start with. It is a crapshoot. The difference between the selective serotonin reuptake inhibitors and the anti-inflammatories is that if ibuprofen doesn't work, you can just try another one. With the selective serotonin reuptake inhibitors, if this one doesn't work, you have to stop taking it and wait a couple of weeks, maybe a month, before you start the second one. So it can take 6 months to a year to get the person on the right dose of the right one where they actually feel better. If that first one didn't work, discontinue it. You got to wait a couple of weeks to start the second one that's going to be trial number 2. During that 2 weeks, you may feel worse, probably will feel worse than you felt before you ever started trying to attack this problem. So it's a messy thing. A lot more on that after the holiday season. So you guys, some of you have a practical. I want to remind everyone that if you're not using your practical grade from this week and you're retaking it next week, that is Monday only. There is no Wednesday practical next week. It is Monday only. I don't know. If I knew, I would tell you. If anybody in the room knows and wants to talk about it, what does Molly have going on? And Friday, is there anything? Over the weekend, anything? Whether it be on campus, whether it be on Facebook, is there anything? Friday, there's an exam 3 review. Here, Facebook, where?
In the lab.
In the lab? Okay, so if you're interested in that, ask one of these people. It seems like some of them know about it. If I knew, I would tell you, but I don't know. She doesn't, have to tell me her schedule.